Hello and welcome, dear students. Today we shall talk about human genome. The main objectives of today's deliberations are to know about the human genome, to try and understand the Human Genome Project, to talk about various genetic markers, and to discuss the organelle genome, and finally to underline the scope and importance of human genome. Genome is just a fancy word for all our DNA which we have inside every nucleated cell. From potatoes to puppies, all living organisms have their own genome. Genome can be defined as the complete set of genes in a cell or living thing. Alternatively, genome or genetic data or genetic makeup is the sum total of all genetic material of an organism which store biological information. The primary function of genome is to store, propagate, and express the genetic information that gives rise to a cell's architectural and functional machinery. In humans, the genome consists of 23 pairs of chromosomes located in the cell's nucleus, as well as a small chromosome in the cell's mitochondria. A genome contains all the information needed for an individual to develop and function. An example of a genome is what determines the physical characteristics of a person. A total amount of genetic information in the chromosome of an organism, including its genes and DNA sequences. The nature of the genome may be either DNA or RNA. All eukaryotes and prokaryotes always have a DNA genome, but viruses may either have a DNA genome or RNA genome. The eukaryotic genome consists of two distinct parts: nuclear genome and organelles, mitochondrial and chloroplast genome. The nuclear genome consists of linear double-stranded DNA. Genome size is typically measured in terms of either mass, usually in picograms, or the number of base pairs it has. Among cellular organisms, the size of genome ranges from less than 10 raised power 6 base pairs, as in smallest prokaryote mycoplasma, to more than 10 raised power 11 base pairs for eukaryotes such as amphibians. The genome of most eukaryotes are larger than those of prokaryotes. In diploid eukaryotes, the characteristic value of haploid DNA content per nuclei is called C-value. It represents the total amount of DNA contained in a single set of its chromosomes, typically measured in picograms. The nuclear genome size of many eukaryotes does not appear to correlate well to the organism's complexity. For example, the genomes of salamanders contain more than 10 times the amount of DNA that is in the human genome. Yet salamanders are less complex than humans. This lack of correlation between genome size and organism complexity refers to the C-value paradox. Therefore, genome size is clearly not an indicator of the genomic or biological complexity of an organism. The human genome is a complete set of nucleic acid sequences for humans encoded as DNA within the chromosome pairs 23 in cell nuclei. Virtually every single cell in the body contains a complete copy of approximately 3 billion DNA base pairs or letters that make up the human genome within its 4-letter language, that's A, T, and GC. DNA contains the information needed to build the entire human body. Human genome include both protein-coding DNA sequences and various types of non-coding DNA that does not encode proteins. The non-coding sequences includes DNA coding for non-translated RNA, such as that for ribosomal RNA, transfer RNA or tRNA, ribozymes, small nuclear RNAs, and several types of regulatory RNAs. It also includes promoters and their associated gene regulatory elements, DNA playing structural and replicatory roles such as scaffolding regions, telomeres, centromeres, and origins of replication, plus large numbers of transposable elements, inserted viral DNA, non-functional pseudogenes, and simple, highly repetitive sequences. Introns or non-coding sequences make up a large percentage of non-coding DNA. Some of this non-coding DNA is non-functional junk DNA, such as pseudogenes, but there is no firm consensus on the total amount of the junk DNA. The human haploid genome content is approximately 3.2 into 10 raised power 9 base pairs. So human genome contains about 21,000 protein coding genes. That's only small part of genome, about 1.5% codes for the proteins of the genome, while as 99.9% is same in all people with differences in that is what makes 0.1% each of us unique from each other. Average gene size is about 27 kilobase in the human genome. Largest gene in dystrophin gene of about 2.5 million base pairs, which codes for a muscle protein dystrophin. Chromosome 1 has highest number of genes with approximately 2,968, while as chromosome Y has lowest number with only 231 genes. Mean exon is about 145 nucleotides. Average number of genes per chromosome is estimated to be about 1,400. Human genome has two types of genomic sequences. One is non-repetitive sequences and another one is repetitive sequences. Non-repetitive sequences consist of sequences that are unique, that is, there is only one copy in a haploid genome. Repetitive sequences, on the other hand, are present in more than one copy. The proportion of the repetitive and non-repetitive varies among individuals. In humans, large fraction of genome is occupied by repetitive sequences. Generally, of the size of repetitive sequences tends to increase with overall genome size. The repetitive sequences fall into two general classes. Moderately repetitive sequences are present in few hundred to several thousand copies in the genome. These sequences generally remain dispersed throughout the genome. A significant fraction of moderately repetitive sequence in human genome is of transposable elements, segments of DNA that have the ability to move within a genome. Sometimes moderately repetitive sequences are multiple copies of the same gene. For example, genes for rRNA and histone proteins, because these are needed in large amounts. Highly repetitive sequences are present in large copy numbers up to millions of copies in genome. Each copy of a highly repetitive sequence is relatively short, generally less than 100 base pairs, often repeated in many times tandemly in large clusters. Because of its short repeating unit, it is sometimes also known as simple DNA sequence. These sequences are often associated with heterochromatin regions of the chromosomes and in particular with centromeres. Highly repetitive DNA in the centromere is called alphoid DNA. Microsatellites and mini-satellites are tandemly repeated DNA. Mini-satellites form cluster up to 20 kb in length with 20 to 50 repeat units. Telomeric DNA is an example of mini-satellites. Microsatellite clusters are shorter, usually less than 150 base pairs. Repeat unit is usually 13 base pair or less. Variable number tandem repeats are types of mini satellites that are exploited in DNA fingerprinting. Now let us discuss organelle genome. Semi-autonomous organelle like mitochondria and chloroplast contains extranuclear DNA. Organelle genomes are usually, but not always, circular DNA which are double-stranded, which code for some but not all of the proteins found in that organelle. Most of the proteins in mitochondria and chloroplast are encoded by nuclear DNA and imported into the organelle from cytosol after they are synthesized on cytosolic ribosomes. Mitochondrial genome is mostly circular double-stranded DNA in humans. All mitochondria contain multiple copies of DNA molecule. The total amount of mitochondria DNA in a cell depends on the number of mitochondria, the size of the mitochondrial DNA, and the number of mitochondrial molecules per mitochondria. Human mitochondrial DNA contains 16,569 base pairs with 37 genes. It lacks introns and contains no long non-coding sequences. So human mitochondrial DNA encodes the two rRNAs found in mitochondrial ribosomes. The 22 tRNAs used to translate mitochondrial mRNA and 13 different polypeptide chains. Human mitochondrial DNA has very little repetitive DNA. All proteins encoded by mitochondrial DNA are synthesized on mitochondrial ribosomes. We humans derive our mitochondria from our mother, that is maternally inherited. Now let us discuss Human Genome Project. So Human Genome Project, which is also called as HGP, was an ambitious research effort aimed at deciphering the chemical makeup of the entire human genetic code, that is the genome. The primary goal of Human Genome Project was to provide researchers with powerful tools to understand the genetic factors in human diseases, paving the way for new strategies for their diagnosis, treatment, and prevention. The Human Genome Project was a coordinated international effort to determine the sequence of the human genome and to identify all the genes it contains. It has produced a plethora of information, much of which is still being analyzed and interpreted. The Human Genome Project, which was completed in 2003, covered about 92% of the total genome sequences. So genomic information has been instrumental in identifying inherited disorders, characterizing the mutations that drive cancer progression, and tracking disease outbreaks. Now let us discuss origins of the project. The publicly funded Human Genome Project began in 1990 under the directions of the James Watson, the co-discoverer of the double helix structure of DNA in 1953. Eventually, the public project was led by Dr. Francis Collins, who had previously led a research team involved in identifying the CFTR gene as the cause of cystic fibrosis. In the United States, the Collins-led Human Genome Project was coordinated by the Department of Energy and the National Center of Human Genome Research, a division of the NIH, that's National Institutes of Health of the United States. It established a 15-year plan with a proposed budget of $3 billion to identify all human genes, originally thought to number between 80,000 and 1 lakh, to map them all and to sequence the approximately 3 billion base pairs thought to be comprised by the chromosomes, 22 autosomes, plus X and Y chromosome, so that is 24 in number. Other primary goals of the Human Genome Project included the following: to establish functional categories for all human genes, to analyze genetic variations between humans, including the identification of single nucleotide polymorphisms or SNPs. Next was to map and sequence the genomes of several model organisms used in experimental genetics, including Escherichia coli, Saccharomyces cerevisiae, C. elegans, Drosophila melanogaster, and Mus musculus, that is the mouse. To develop new sequencing technologies such as high throughput computer automated sequences to facilitate genome analysis. Further, to disseminate genome information among both scientists and the general public for larger good. Recognizing the impact that genetic information would have on society, the Human Genome Project also set up the ethical, legal, and social implications, that is ELSI program, to consider ethical, legal, and social issues arising from the Human Genome Project and to ensure that personal genetic information would be safeguarded and not used in discriminatory ways. As the Human Genome Project grew into an international effort, scientists in 18 countries were involved in the project. Much of the work was carried out by the International Human Genome Sequence Consortium, involving nearly 3,000 scientists working at 20 centers in 6 countries, that is China, France, Germany, Great Britain, Japan, and the United States itself. In 1999, a privately funded Human Genome Project led by John Craig Venter at Celera Genomics, aptly named from a word meaning swiftness, was announced. Celera's goal was to use whole genome sequences, that is WGS, and computer automated high throughput DNA sequences to sequence the human genome more rapidly than the Human Genome Project had proposed. Whole genome sequences, that is WGS, is a comprehensive method for analyzing entire genomes. The public project had proposed using a clone-by-clone approach to sequence the genome, which was a very laborious process. Recall that Craig Venter and his colleagues had proven the potential of whole genome sequences in 1995 when they completed the genome for Haemophilus influenza. Celera's announcement set off an intense competition between the two teams, that is the public and the private funded organizations, which both aspired to be first with the human genome sequences. This contest eventually led to the Human Genome Project finishing ahead of the schedule and under budget after scientists from the public project began to use the high throughput sequencing and the whole genome sequencing strategies as well. Now let us discuss the major features of the Human Genome Project. In June 2000, the leaders of the public and private genome projects met at the White House in the United States with President Bill Clinton and jointly announced the completion of a draft sequence of the human genome. In February 2001, they published an analysis covering about 96% of the euchromatic region of the genome. The public project sequenced euchromatin portions of the genome 12 times and set a quality control standard of a 0.01% error rate for their sequences. In 2003, sequencing was completed and the work was sufficient to pass the international project's definition of completion. We now understand that there are approximately 3.2 billion chemical base pairs arranged in precise sequence with nearly 21,000 genes in human genome. Major features of the Human Genome Project are summarized as: The human genome contains about 3.2 billion nucleotides, but protein coding sequences make up only 2% of the genome. The genome sequence is 99.9% similar in individuals of all nationalities. Single nucleotide polymorphisms or SNPs and copy number variations that in CNVs account for genome diversity from person to person. The genome is dynamic. At least 50% of the genome is derived from transposable elements such as line and ALU sequences and other repetitive DNA sequences. The human genome contains approximately 21,000 protein coding genes, far fewer than originally predicted, that is the number of 80,000 to 1 lakh genes. The average size of a human genome is 27 kb, including genes, regulatory regions, introns, and exons. On average, mRNA produced by human genes are 3,000 nucleotides long. Many human genes produce more than one protein through alternative splicing, thus enabling human cells to produce a much larger number of proteins, perhaps as many as 200,000 from only 21,000 genes. More than 50% of the human genes show a high degree of sequence similarity to the genes in other organisms. However, more than 40% of the genes identified have no known molecular function. Genes are not uniformly distributed on the 24 human chromosomes. Gene-rich clusters are separated by gene-poor deserts that account for 20% of the genome. These deserts correlate with G bands seen in stand chromosomes. Chromosome 19 has the highest gene density, and chromosome 13 and the Y chromosome have the lowest gene densities among all the chromosomes in humans. Chromosome 13 contains the largest number of genes, and the Y chromosome contains the smallest number. Human genes are larger and contain more and large introns than genes in the genomes of invertebrates, such as Drosophila. The largest known human gene encodes dystrophin, a muscle protein. This gene, associated in mutant form with muscular dystrophy, is 2.5 MB in length, larger than many bacterial chromosomes. Most of the gene is composed of introns. The number of introns in human genes ranges from 0 to 234 in histone genes and in the genes for which it encodes, that is the muscle protein. Two of the biggest surprises discovered by the Human Genome Project were that less than 2% of the genome codes for the actual proteins, and that there are only around 21,000 protein coding genes, that the number of the genes had originally been estimated to be about 1 lakh, based in part on a prediction that human cells produce about 100,000 proteins. There is still no consensus among scientists worldwide about the exact number of human genes. One reason is that it is unclear whether or not many of the presumed genes produce functional proteins. Currently, annotation predicts that the human genome encodes approximately 21,000 proteins, but due to post-translational modifications and other processes, the total number of proteins present in human cells is thought to be anywhere from approximately 200,000 to 1 million. The number of genes is much lower than the number of predicted proteins, in part because many genes code for multiple proteins through alternative splicing. Alternative splicing patterns can generate multiple mRNA molecules and thus multiple proteins from a single gene through different combinations of intron-exon splicing arrangements. During the Human Genome Project, functional categories were assigned for human genes, primarily on the basis of functions determined previously, for example, from recombinant DNA, cloning of human genes, and known mutations involved in human diseases. Comparisons to known genes and predicted protein sequences from other species. Predictions based on annotation and analysis of protein functional domains and motifs. Although functional categories and assignments continue to be revised at the time of Human Genome Project was completed, the functions of over 40% of the human genes were unknown. Determining human gene functions, deciphering complexities of gene expression, regulation, and gene interaction, and uncovering the relationships between human genes and phenotypes are among the many ongoing challenges for genome scientists. The Human Genome Project originally revealed that in all humans, regardless of racial and ethnic origins, the genomic sequence is approximately 99.9% the same. Most genetic differences between humans result from single nucleotide polymorphisms, that is SNPs, and copy number variations, CNVs. The genome is not static. Genome variations include the abundance of repetitive sequences scattered throughout the genome. Verify that the genome is dynamic, revealing many evolutionary examples of sequences that have changed in structure and location. SNPs, or single nucleotide polymorphisms, are single base changes in the genome, and variations of many SNPs are associated with disease conditions. After the draft sequence of the human genome was completed, it initially appeared that most genetic variations between individuals of 0.1% differences were due to SNPs. Copy number variations are, on the other hand, duplications or deletions of relatively large sections of the DNA on the order of several hundred or several thousand base pairs. Although most human DNA is present in 2 copies per cell, that is the diploid, one from each parent, that is male and female or father and mother, the CNVs are segments of DNA that are duplicated or deleted, resulting in variations in the number of copies of a DNA segment inherited by individuals. A majority of human genes are similar in sequence to genes from closely related species such as chimpanzees. In many ways, the Human Genome Project has revealed just how little we know about our genome. It's now possible to access databases and other sites on the internet that display maps for all human chromosomes. The Human Genome Project most valuable contribution will perhaps be the identification of disease genes and the development of new treatment strategies as the result. The omics revolution has created a new era of biological research because of the Human Genome Project. The Human Genome Project and the development of genomics techniques have been largely responsible for launching a new era of biological research, the era of the omics. It seems that every year, more areas of biological research are being described as having an omics connection. Some examples of omics are, for example, proteomics, the analysis of all the proteins in a cell or tissue. Then the metabolomics, the analysis of proteins and enzymatic pathways involved in cell metabolism. Third, glycomics, the analysis of the carbohydrates of a cell or tissue. Fourth one, toxigenomics, the analysis of the effects of toxic chemicals on genes, including mutations created by toxins and changes in gene expression caused by toxins. Fifth, metagenomics, the analysis of genes of organisms collected from the environment. 6. Pharmacogenomics, the development of customized medicine based on a person's genetic profile for a particular condition. The 7th one, that is transcriptomics, the analysis of all expressed genes in a cell or tissue. Dear students, with this, we come to the end of this module. Hope you have understood it well. See you again with another topic. Thank you and goodbye.